Hi everyone, this is a demo of the Benchling platform. So here you can see that if you create an account, you'll get access to different tabs that allow you to create projects. And here, create a project as an example. And here you can create files, folders, entries, protocols for your experiment. You can also upload your DNA or RNA sequence or an amino acid sequence of your choice. So today we're going to do an example using different sources of tools. So here we have a genome browser, which allows us to look up our genes of interest and to derive the RNA sequence as well as the protein sequence. We're also going to be using the BLAST tool in order to look for the origin of our protein or of our nucleotide and identify the organism that share the sequence. We're also going to look at the protein further using UniProt, as well as a tool called the Human Protein Atlas, which allows you to look at staining of this protein, to look at what does it look like in a natural human body, and what are the profiles associated with that. Finally, there's also a site called AdGene, which is really helpful for looking for plasmids that are expressing a gene of interest. So let's say you want to find the sequence of a fluorescent protein and it's a synthetic sequence. You can go under fluorescent proteins and then you can browse for your protein of interest. And you can see there's different applications that are categorized, which is really helpful in finding a plasmid that fits your need. Okay, so today we're going to use MILD as an example. So this is a gene involved in myogenic differentiation, in another word, skeletal muscle differentiation. Once you search up your gene of interest by its name, you can do it by the short form MILD, and then click on the sequence, which will then take you to a card like this. And here you can either open the mRNA sequence or the protein sequence. So here I'm going to open the protein sequence. And you can see that all of these are expressed in amino acids. And you can see the length as well, which is 320 amino acids. We're now going to upload that sequence to Benchling. So under AA sequence, you can do new AA sequence. Here we're going to label it as MILD. You can select the schema if you want to organize your entries. Okay, then you just paste your sequence here. And now you can visualize all of your amino acid sequences. It also tells you some properties about them, which may be helpful to you as well. We're going to select everything and back translate. So then you can see if this gene came from humans, you can select Homo sapiens as the organism. You can also choose whether you want to match the codon usage, meaning that whatever codon is shown here will be what will be translated back, or use the best codons. Sometimes there are more frequent codons that are used in a target organism, such as in humans. So in our case, it doesn't really matter. We're just showing that there's an option. We can just choose to use best codons. Preview optimization, and then save as new sequence. And here we go. So now we have our DNA sequence, and we can see that it starts with an ATG, which is the start codon to any sequence, and it should end with a stop codon as well. And if it doesn't have a stop codon, then you want to make sure that you add it in before you use this for downstream applications, such as expressing it in a cell of interest or cloning it for bacteria purposes. But if you wanted to just use it as a gene fragment, then the start codon is what you need. Okay, so now that we have this sequence, this is the cDNA of this gene, which is different from the genomic DNA. So in a separate talk, we'll talk about the differences between the genomic DNA and the cDNA. Here, this is the sequence that is needed to express your gene of interest. So now that we have the sequence, we can order this either as fragments and put them together in the lab, or we can use it for downstream in silico analysis. So then I'm going to show an example where let's say that we don't know our the sequence name or where it came from. Let's say this is a file that we get and we don't know what the gene name is. What you can do here is you can select everything and create a translation. So let's say we do a forward translation. We can put unknown here because let's just pretend for a second that we don't know what this is. Okay, so now you'll see that it shows you the alignment of the protein sequences as well, with the first codon being methionine. So now with this translated sequence, we can copy our translation. So here, when you select copy, you can select an amino acid translation or copy the DNA sequence. So for this example here, I'm going to copy the protein sequence. So now we can go into the alignment tool, which consists of nucleotide blast or protein blast. I'm going to select protein blast, paste in our protein of sequence, and then blast it. This will take a little while. So while we're doing that, we're going to go into the human atlas to look at the protein further. Okay, so let's say we want to search up ILD. Great. So now we have the gene description, as well as whether it's expressed in tissues, and if so, what types of tissues. So if we click into that, we can visualize the protein as a 3D structure right here. And if you're doing any protein engineering work, this will be more relevant for you. But it's always good to know in 3D what your protein actually looks like. You can also look at the protein expression, and here you can see that it is predicted to be an intracellular protein, and it is localized to nucleoplasm. We can see that it's enriched in skeletal muscle, which is what I had mentioned earlier, and it is not detected in the brain, and you can look at other cell types to make sure that, whether it expresses or not, but it shouldn't express in the brain, the blood, anything like that. What's really helpful here is that you can also see if it's related to any cancerous conditions. You can also get a detailed overview of the protein function, which is also really helpful for you to learn more about what this protein does. Okay, now let's go back to the BLAST tool. Now we have the analysis finished. You can see that it gives you a whole bunch of outputs, and it shows you what cluster ancestor it came from. So because this gene is commonly found in many organisms that have skeletal muscle, because it regulates the differentiation, a key step in muscle generation, it will be found in many different species. And you can see that when you look at similarities, you can identify how similar the sequence that we had inputted is to the sequences found natively in these animals. And here you can see we do have a human version that's identified here. It's also found in rodents, crop-eating macaques, and yeah, many other organisms, which is really interesting. So if you didn't know the protein that you have here, we'll have to log in. So let's go back to here. So yeah, so if you didn't know which protein your gene of interest belongs to or which organism it came from, this is a really good way to identify the source and the identity of your region of interest. All right, and that's it for the intro to Benchling and using the tools associated to help you identify a gene and translate to protein and the reverse process as well. And stay